My name is William Bauer. I'm a high school science teacher, OpenSciEd facilitator, and curriculum writer. Lesson 9 is a fun lesson because it provides students some opportunities to get creative with their design of their experiment with UV sensitive yeast. More about that later. Let's review how we got here. In Lesson 8, we looked at inherited risk factors for cancer. We figured out how mutations that increase the risk of cancer can be inherited in families. During meiosis, chromosome pairs can swap regions of DNA in a process called crossing over, and new allele combinations can form, like seen in the pedigree to the right. In Lesson 9, we turn our attention to our questions about how genes interacting with the environment impact who is at risk of developing cancer. This lesson requires a significant amount of prep time because we are going to be using two different types of yeast for an investigation in the middle part of this lesson. I'll go into more detail about the lab setup later. There are three big sections of this lesson. Let's dive into what's happening in each. In the first section, parts 1 through 3, students revisit the class consensus model and ask new questions about potential mechanisms related to environmental risk factors for developing cancer. There are a lot of environmental mutagens to investigate, but this lesson will focus on UV radiation and skin cancer risk. We begin our more specific focus on skin cancer by looking for patterns about skin cancer data organized by location within the United States and race and ethnicity. We figure out that white people tend to have more skin cancer when they live in sunnier areas. It's not clear what is happening for the Black or Hispanic populations as the amount of UV radiation goes up. In the middle part of this lesson, we design and conduct the yeast investigation. Students will expose wild type, aka normal yeast, and UV sensitive yeast to UV radiation. They also design a plan to test the effectiveness of sunscreen, clothing, at protecting the yeast from UV exposure. From the lab, we figure out that the more UV sensitive yeast die after UV exposure because they lack a gene to repair UV damaged DNA. In the final section of this lesson, students read about the mechanisms behind UV exposure and cancer risk. We figure out that UV exposure causes DNA damage. Melanin and protective measures such as sunscreen help reduce this damage. After looking at the skin cancer data, conducting the lab, and reading about the mechanisms behind skin cancer risk, we figure out that UV radiation can cause mutations in DNA. People in sunnier areas with more UV exposure have potentially more mutations. These mutations can cause changes in protein structure and function that lead to cancer. The skin has a protective mechanism against damaging UV radiation. That is governed by a protein called melanin. Variations in exposure to UV and variation in melanin production can lead to different levels of risk in the human population for skin cancer. Here are some key moments. In part 3, investigating skin cancer data, we brainstorm questions about environmental factors and cancer risk. We narrow our focus to skin cancer by looking at data like the graphs pictured on the slide. As my students looked at these graphs, they tended to overlook the range of numbers on the y-axis. You might miss it too. Notice that the range for the graph about the white population, the range goes from 15 to 45 per 100,000. But if you look at the range for the Black population, the range is from 0.2 to 1.5. Those are significant differences in the range. Your students might be like mine and focus mainly on where the dots fall on the plot, but not pay attention to the numbers that are represented on the y-axis. when they look from one graph to the next. In Part 5, students design their investigation with the yeast. We make an analogy between human cells and yeast cells. If the yeast cells die after UV exposure, that would be like someone getting skin cancer from UV exposure. Students identify which factors they want to test, like the amount of light exposure or environmental factors that might protect against UV damage. You can see examples of these variables in this table below, taken from the teacher's guide. Encourage students to try a variety of environmental protection factors or different intensities of light. This is an opportunity for them to get creative with their experimental design. Remind students that whatever we try, we should conduct the test with the normal wild-type yeast and the UV-sensitive yeast. By the time we get to Part 12, we are ready to share our results to see how the normal and UV-sensitive yeast responded to different ways of protecting yeast from UV exposure. To close out this lesson, we dive into mechanisms behind the patterns we've noticed related to UV exposure and skin cancer. What exactly does UV exposure damage in the DNA? In the reading, students find out that UV causes our cytosine nucleotide to change to thymine. This is a good opportunity to revisit what students learned about DNA, mRNA, and proteins in lessons 5 and 6 of this unit. Switching a C for a T in a gene like P53 could lead to skin cancer. It's important to emphasize that melanin and sunscreen can lessen the impact of UV radiation on our skin. At this point in the unit, we have a really deep understanding of how complex the 3 different mechanisms for skin cancer risk are: random mutations, inheritance, and now in Lesson 9, we've added on environmental factors as part of our explanation of who gets cancer and why. We'll put all those pieces together in the next lesson. Here are some tips and tricks for this lesson. This lab requires you to do some prior planning. If it's your first time through this unit, it might be challenging to predict when exactly you'll reach Lesson 9. Go easy on yourself. But you should know that you want to order your kit so it arrives 3 to 4 weeks prior to the class investigation. The kit contains all the materials except for the live yeast. The kit contains instructions for how to order the live yeast. You should order your yeast so that it arrives 1 to 2 weeks before the investigation. You'll also need to pour your plates for the yeast to grow on 4 to 7 days before you're planning to do the lab. You should also try to determine how exactly you're going to expose the yeast to UV. When you're thinking about that, consider the time of year, location, and weather as important factors. I've had success with an indoor grow light, as I am usually conducting this experiment in the winter in the rainy Pacific Northwest. A few more tips and tricks. I like to let students get creative in selecting different ways to protect the yeast from UV exposure. Give them control on how to design this lab so that they can investigate their questions about skin cancer and UV exposure. Try a variety of different fabric of different colors and thickness and materials, or different sunscreens. I even had a student try using tanning oil to see if that would make the UV exposure worse. Why not? This is science. Try it out. If students have a lot of ideas or you're limited by the amount of materials, you can always divide the petri dish in half, where one side is protected and the other side is unprotected. Another important tip when you've done the lab and you're now interpreting the results, make sure you're clear about the analogy between the lab results and skin cancer in humans. Because we normally associate more cell growth with cancer, it can be confusing. The results can be confusing for students and they can misinterpret the results. More growth means the yeast cells are not affected by UV. Less growth equals the yeast cells are damaged by the UV. This can be confusing because we normally associate more cell growth with cancer. My students confused this the first time. In terms of vocabulary, the only word we're really encountering in this lesson is melanoma, aka skin cancer. At the start of the lesson, you'll refer back to both the class consensus model and the driving question board to surface ideas and questions students have about the environment and cancer. My students had a lot of questions for this lesson. Thanks for listening. In the next lesson, we'll put all the pieces together and take a transfer task in lesson 10 as we answer the question, who gets cancer and why?